Well, first of all, thanks a lot for the invitation to come speak here today. We have a number of different projects with JGI, all involving sequencing to some extent. And so some of these are involved, we're looking at soil, we're looking at beetles, we're looking at plant litter decomposition, but they all revolve around looking at microbes decomposing plant polymers to some extent. And Eric gave a great introduction to this emerging field of community systems biology. And today I'd like to add another component to the community systems biology, and that's biochemical conformation of function. So we're working on a project in a grassland in Southern California, which I'll introduce in a few minutes. And it involves a range of different activities, including modeling of microbial activities. And our role in this is to provide biochemical conformation of specific functions related to plant polymer decomposition. So why are we interested in this? Well, we're interested in how specific ecosystem processes will change as a result of predicted global change. And here I'm just showing two examples that we're particularly interested in. If you look at, this is a prediction of projected change in precipitation over the next century. You can see that across the US, but particularly in Southern California, we're predicting increased dry periods in winter and more so in spring into summer. So extended drought periods and decreased frequency of precipitation is likely to impact these regions of California. These regions, and also in other parts of the US, but particularly in California, due to urbanization and agriculture, has significant problems in atmospheric N deposition. And this can really have a profound impact on ecosystem function in many different ways. Now, both these processes, these changes, can affect microbial composition and microbial activity directly and indirectly. So drought and nitrogen deposition can affect the physiology of organisms. It can affect the succession of organisms directly through the interaction of osmotic stress or changing nutrient stoichiometry, but it can also affect the function and the activity of microorganisms through changes in plant species composition. And in some cases, these factors can counteract each other. So it's very difficult to try and parse out the indirect versus indirect activities without a framework. So we're working at a site called Loma Ridge, which is operated by UC Irvine in Southern California. This has been operated by Mike Golden from UC Irvine. Mike has been running this site for about a decade at this point, and he's been looking at rainfall and nitrogen manipulations. It's a Mediterranean grassland, so we've got relatively cool, wet winters, dry summers. And the primary plant productivity period is somewhere between September, November, up to typically about April, when plants senesce, with peak plant productivity around about January or February. Okay. So we're taking what we could call a trait-based framework. And this is an intermediate modeling activity which is sort of in between the standard biogeochemical models and something like a genome-scale metabolic model. So a genome-scale metabolic model, you're considering all of the functions within a cell and how those functions interact. It's extremely complicated to upscale that to a microbial community. A biogeochemical model represents, if it's an elaborate biogeochemical model, it might represent enzymes, but these are usually parameterized by empirical observations. And a trait-based model takes the physiological and biochemical capabilities of microbes, represents them as traits, much like the plant community represent traits, leaf area index, things like that. So we're representing different traits of microbes, and the different types of traits we're representing relate to their activity, their function. So those functions could be the ability to depolymerize a specific polymer, to take up the monomers from that, and then other traits, so they're related to the kinetics of those processes, so presence or absence of that capability, the kinetics of the process, the kinetics of growth, etc. So stoichiometry of biomass, things like that. And also other traits in terms of environmental response. So whether an organism is drought tolerant, drought susceptible, whether it increases due to nitrogen addition, something like that. So these are what we'd call environmental response genes. And ideally, we'd like to be able to identify these, the linkage between them, some taxonomic markers, and be able to relate the environmental impact on the microbe through the environmental response genes and the effect of that on the function of the organism through the linkage of the functional genes to the environmental response genes. So this requires functional characterization. It also requires assembly. I can't stress that enough, to link these traits together. And ideally, then, we'd like to be able to predict the expression of these traits and relate that to function in terms of a decay rate. Okay. Now, I'm not going to go through all of this modeling today. This is work that's really driven by Steve Allison, and these are my co-PIs on the project. But I'm going to give some examples of the work that we do to help parameterize and constrain these models. So Steve Allison has developed a model which he calls DEMENT, and for multiple reasons. But this is the decomposition model of enzymatic traits. And this is a trait-based model, so it explicitly represents microbial function. And that may seem like it's obvious to this community here. But in biochemical modeling, microbes are more often not represented explicitly, or represented by a black box, or represented by their biomass with some generic rate constants that represent an average among microbes, which is obviously not accurate. So in this case here, in this model, it's very simple. Microbes are represented by their stoichiometry. They have the capability of producing specific extracellular enzymes, capability of taking up the products of those enzymes through specific and general transporters, and substrate use efficiencies, which result in variable fractions to biomass versus respiration, etc. Now, you can represent each of these microbes in a simplified form in two-dimensional space. Populate a grid. This grid represents the surface of a leaf here. And on the surface of the leaf, you have you set up this model so that each of these grids has a likelihood of being populated by different microbes, and then you allow the system to emerge. So you run it forward in time. You look at emergence of different microbial guilds depending on their function or their linked traits. So in the model setup, this is just an example, and this has been published by Steve. Taking 100 taxa, we don't know what they are, this is just a simulation. A 100 by 100 grid. Taxa may possess 0 to 20 enzymes. Those enzymes are related to 12 chemical substrates that are a proxy for fresh plant litter. And then each of those substrates is degraded by at least one enzyme, but some enzymes can have multiple substrates. Okay. So you set this up. You've got a matrix here of taxa by enzymes, and then enzymes by substrates. And these can be randomly parameterized, so drawn either from a distribution where you have got some information about the distribution of these genes, so you can take either a range or a probability distribution function, and you can sample from that. But this can either be randomly parameterized, or else it can use specific information from an environment. I'm going to talk about some of that. Okay, so when you randomly assign these traits, what you see is these guilds of organisms emerge. So guilds being organisms with similar traits with similar responses. This just gives an example here. If you look at the blue colors are taxa with more enzymes. So between the 0 to 20, these are organisms that were seeded with more enzymes. What you see is that they reach a higher maximum, but they reach a maximum later. So there's a cost associated with that, cost associated with more enzymes. Now, key to trait-based modeling is the accurate parameterization of trade-offs. So if you don't have a trade-off between the possession of one trait and another trait, let's say, for example, you've got many enzymes. If you have many enzymes and a very high growth rate, so no cost associated with many enzymes, you get what's called a Darwinian demon. And these Darwinian demons are organisms that are optimized for every possible trait. And we know that in nature that does not occur. These organisms do not occur. They're hypothetical because there are always trade-offs. Energy is finite, and energy needs to be allocated. So there are trade-offs. So parameterizing those trade-offs, identifying those trade-offs is very important. But in this case here, you can see this is a simulation, and this just shows, I don't know if you can read these, but these are the various substrates. So you've got cellulose, lignin, degrading. So this is a decomposition pattern over time simulated with these. Okay. And this is an exponential decay, roughly approximates what happens, but and over a time frame here. So this is with relatively static environmental conditions, and over time we get the decay of this, a plateau which roughly corresponds to, you're left with about 20% of mass or something like that, and then you get an increase in microbial byproducts, dead microbes, and then they become substrates, etc. Right? Okay. So that's with random assignment of traits. But we know, if you just go to sequence databases, that traits are not random. So extracellular enzyme trait distributions are non-random. For example, the capacity to produce extracellular enzymes can vary at very fine phylogenetic scales. Very similar organisms, phylogenetically, can have different enzyme distributions. We can also see that, a wide range of organisms may have the potential to degrade cellulose. An even wider range have the potential to cozy up with cellulose degraders and produce beta-glucosidase to take the product of cellulose degradation. But these are not randomly distributed, so they're unevenly distributed around the tree. Right? Okay. So why would we take why would we use random simulation? Just basically to test models. Okay. When you do the random simulations as well, you also identify possible trait combinations that might result in fitness of an organism under specific conditions, and that provides hypotheses then to go and test. But in individual environments, as Eric just showed, the selective pressure and the life history of that environment may have selected for different trait distributions. So each individual environment may not represent these patterns that we see in sequenced genomes. So we're taking an approach to try and get at this functional diversity using functional metagenomics. And this is just an overview of the different options available in functional metagenomics. There are others. So we take a sample, and you can extract DNA and RNA from the sample. In this case here, extraction of metagenomic DNA, then to functional screening, RNA sequencing, then potentially functional screening of transcripts, extraction of DNA, shotgun metagenomics, gene annotation, functional prediction, and then synthesis of those genes and optimization of expression signals. And this is something that I know Jay Jia is very interested in the moment, and Sam Deutsch is going to talk about on Thursday. This is maybe represents somewhat of the future of this approach. For now, we've taken this approach here, where we've extracted DNA, created libraries, screened those libraries against various substrates. And what that gives us is a confirmed biochemical activity. We're not just relying on annotation in this case. We have an actual activity. And then really the power of this is relating this to this, and then eventually looking at something like proteomics to confirm expression and activity of these genes. So in this site in Loma Ridge in California, we have different treatments. We have our ambient treatment. So this is plant litter that has fallen on the ground, either in litter bags or just naturally. We have a nitrogen-enriched set of plots, 8 plots for each, and reduced precipitation. And we pool from each of these 4 plots into a subsample, 4 plots into a subsample. We sampled at different times during this decomposition trajectory, December, February, September, March, and we created phosmid libraries using the Epicenter copy control phosmid library kit. Okay. So we've taken our genomic DNA, we've got about 45 kb inserts into the phosmids, and we end sequence those just to see what the diversity of the inserts is like. And this is dominated by proteobacteria, actinobacteria, Bacteroidetes, and fungi. So at least these genes are being maintained in E. coli. There's nothing toxic in those at the moment. When we compare this to just random shotgun metagenomic profiles, the phylogenetic profiles are extremely similar to that. So we don't think there's a significant bias in this at this point. Okay. So we have a range of substrates that we want to screen these libraries against, targeting cellulose degradation, hemicellulose degradation, starch, chitin, lignin, phosphatases targeting organic phosphorus, and protease. Okay. Now, the issue is that we're using this E. coli from this kit, and this E. coli also has activity against some of the substrates, and that reduces our sensitivity. And what you can see in these plots here is that there are some obvious positives here, but then there's a general background. We're looking at a kind of a light blue tinge all the way along here. So we need to reduce this background to improve the sensitivity. So we knocked out these genes involved in starch, in alpha amylase and alpha glucosidase, and in phosphatase, alkaline phosphatase. Okay. So we've got to get reasonable coverage of this microbial community, we have to sequence we have to screen a lot of clones. And we're also screening against a lot of substrates. And this very quickly multiplexes into an enormous challenge. So we came up with a strategy of multiplexing our substrates based on activity. So we multiplex substrates for cellulose degradation, targeting endocellulase, cellobiohydrolase, beta-glycosidase, hemicellulose degradation, endoxylenase, beta-xylosidase, starch, chitin, peptide breakdown, phosphate release, lignin degradation, etc. And we were able to multiplex these because the detection method for each of these is distinct so that they can be combined in the same assay. So we've either got absorbance or fluorescence, or in some cases, these are just on plates. We have visual inspection of halos on plates and automated picking. Okay. So this allows us to screen for many, many different substrates in fewer assay vessels, I guess, in fewer wells of a plate. Okay. The other thing we need to do, we expected that based on the proportion of genomes that contain these traits, we knew that our hit rate was going to be relatively low. Even assuming if every organism in the sample had these traits, just based on gene frequency in genomes, we knew our rate was going to be low. So we came up with a pooling strategy where we pooled 16 different phosmid clones into each well of a plate. We screened those for activity. Wherever we found a positive, we then demultiplexed. So we knew which wells went into that single well. We screened every single one of those. And then this results in, I just put some numbers up here, but $93 per 1,000 clones in our lab, as opposed to over $1,000 for. So that allows us to screen many more. Okay. So we worked with folks at the Joint Bioenergy Institute. We used the robotics facilities there to do automated picking of the phosmids to 96-well format. These were then arrayed into different substrate plates. They were screened for activity in liquid media using, as I said, fluorescence and absorbance substrates. We also did the same thing. We took our colony pickers, arrayed the plates on large plates, the 96-well plates on large plates, looked for activities there. These were either automatically or manually picked. And then sequencing the positive clones to identify which organisms are these traits associated with, and which organisms contain multiple traits. Right. Okay, so overall, to date we've screened over 300,000 clones. That represents about 12 gigabases of DNA. That's roughly 2,000 bacterial genomes screened for activity. We screened 14 different conditions, 13 substrates, and phosphate at 2 different pHs. And overall, that's about 4.4 million assays. And you can see our hit rates. These are hit rates for our different substrates along here. Our highest hit rate was for beta-xylosidase. But these are variable, all extremely low. Some of these things, the chitin, azure, this is depolymerization of chitin polymers. We didn't see any activity there. And we didn't see any activity for our oxidative enzymes. And we can maybe discuss that later. But I will say that we do see expression of some fungal genes in there, but not the oxidative enzymes. And in other projects, we do see expression of fungal oxidative enzymes. So that's a story to be worked out still. At different times, in libraries from different times, we have different hit rates, et cetera. So we want to combine this information. So we have these phosmids. We have confirmed activities for each phosmid. We want to know where those phosmids came from, what genes are on those phosmids, and maybe associate different phosmids with a single organism. Multiple phosmids may come from a different organism depending on where those genes are associated on an organism. So we take our confirmed positives, array them into plates, DNA extraction, index those, and fragment using Nextera, sequence them at MySeq 2 by 250 base pairs so that they overlap. So we end up with somewhere between 400, around about 400 bases of quality sequence. Demultiplex those, assembly, do gene calling, and some initial annotation using HMM models. From the assembled phosmids, we want to bin those. There are many ways of doing this. Our first pass has been tetranucleotide frequency binning. So that's associating different phosmids with a specific genome bin, trying to identify whether trait linkage occurs. Okay, so in our positives, we've identified that the majority of them are coming from alpha proteobacteria, but we've also got beta proteobacteria, gamma proteobacteria, actinobacteria, Bacteroidetes, eukaryota. Most of those are fungal. And we can see that we've got representation of different glycoside hydrolase families, carbohydrate binding modules, etc. And here in comparison to what you would get from sequenced isolates, the representation of these organisms, we can see an enrichment of GH3 and GH5 of the cellulase active families relative to sequenced isolates. Okay, so there's already a different view of the distribution of these activities in our environment versus what you would see in the database. Okay. So far, we've just sequenced 279 phosmids of about 800 that are still in progress. Okay. So to link phosmids to genomic origin, you can use various binning methods that Eric outlined. And again, this is just tetranucleotide frequencies, but we're also using coverage, and coverage over time, and coverage over time by experimental manipulation. That creates variability in your data set, variability in response trajectories of different organisms, and you can use those response trajectories to further refine your bins of potential genomes. So we're using tetranucleotide frequencies and then organizing this using self-organizing maps in this approach that Greg Dick from Jill Banfield's group pioneered. So if you look at the phylogeny of these bins, we have these bins. And these are not full-scale genome bins, of course, because we're just we're assigning phosmids to genomes. We would ideally we'd like to be able to take the shotgun metagenome data from this, the assemblies from the shotgun metagenome data for this, and also co-bin that with these phosmids that have confirmed biochemical function. So you can see that we have a lot of representation of alpha proteobacteria, particularly within the Sphingomonidaceae. These are distinct. We've characterized these based on ribosomal proteins and other conserved proteins. And these are distinct bins that contain distinct organisms. It's possible that they can be parsed further, but for now, this is our resolution. So we've Sphingobacteria, Xanthomonidaceae, Actinobacteria, Mycococconae. We've Ascomycetes, some Bacteroides, two different types of Bacteroides, some beta proteobacteria, all contributing potentially to plant polymer decomposition. If we color these maps in a different way, we can look at how different bins are enriched at different sampling times. And one thing you'll notice here is that our sampling time in March, that we see these bins are relatively overrepresented. So the Actinobacteria and the Ascomycota were more abundant in our March samples. This is just, we're just browsing data here at the moment. Okay. We can look at our experimental treatment. We have our control, our reduced precipitation, and our nitrogen effect. Not so much of a pattern, okay, at this level. Maybe some clusters here and there that look like they might be enriched. These may be, further refined genome bins, but you get the idea. Okay, so we have our phosmids. We've assigned our phosmids to specific bins. And for each of the phosmids, we have biochemical activities. So these are our biochemical activities. Each line in this is a phosmid, and then these are marked by their bins. Okay, so you can see these are traits, whether an organism can produces cellulase, hydrolase, etc. So we can start to put these things together in terms of what is the potential role and what is the niche of each of these organisms. As I said, we're only about a third of the way through our phosmid sequencing, so we've more information to add to this, plus adding the shotgun genomic information to this. But it does tell us that not every organism has every function, and that the distributions of those functions vary depending on the organisms and on the bins. Okay. Again, if we look at the characterized glycoside hydrolases and laccases, just look at the pattern. The first pattern here is basically we take all of the bins, and this is the distribution of these functions in our system, based on phosmids that had activity to these substrates. So we've got potentially in each of these bins we've got trait linkage. So this is we've got confirmed activity, and then we know that these genes are present in these phosmids, and you can see that the pattern changes quite significantly across different bins. Right? So we have this genomic resource with confirmed biochemical activity, or biochemical activity. And we can take samples from the environment, taken at greater frequency, and map those back to these phosmid bins. And we verified these phosmid bins, or we've identified that they have specific functions. So we sampled, or rather Renaud and Adam sampled, and sequenced 100 base pair paired-end reads, about 11 million reads per sample, roughly, from 8 different time points. And those time points represent 2 seasons, but on litter that was decomposed from that died in the first season, basically. So we begin here at April, just around about after plant senescence. First sampling here, then sampled in I think April, August, June, September, December, and March. There's a February in there somewhere as well. Okay. So we have all of these short reads, and then we can map these short reads back to the bins and look at how the coverage of different bins changes over time or with treatment. And the first thing we noticed with these bins is that the fungi and bacteria have opposing seasonal responses. So the fungal bin, let me just jump back here. This is precipitation. This is rainfall. So just the seasonal variation in precipitation. We're in a Mediterranean grassland. We've got a wet winter and a dry summer. Okay. So bear that in mind. When it's wet, bacteria are higher, relatively high. When it dries down, here we have it in August after a summer of drought, relatively dry conditions, all bacteria decrease, the fungi increase, etc. So we have this opposing response. And this bar here is actinomycete. These are the fungi. And if you look at these, their correlation is negative 0.88 or so. Right? So they have opposing responses. What's important about this is that we know the different functional potential of each of these different bins. And so we'd like to be able to relate that to the decomposition processes. Now, we're not there yet, but this is where we're going with this data. Okay. What we can see when we look at the differences in treatment effects, we've got different bins. Here are 2 bins. This one is a sphingomyelinaceae. Here's another sphingomyelinaceae, very related by ribosomal protein, and with different environmental responses. These ones are relatively drought tolerant and N insensitive. More or less follow the same pattern as the control. And these ones are drought susceptible but N insensitive. You see here the very large decrease. And this drought susceptible is really due to this response trajectory following drought. So we've got a dry condition, and then a drought treatment overlaid on the seasonal variation results in a different trajectory than when water is not excluded or not reduced. Okay. And then we have the ascomycetes, which we would call drought tolerant. They increase under dry conditions, but N sensitive. And you can see that they have a different response trajectory when grown under elevated N conditions. So what we'd like to do is be able to relate this information back to, we have the trait distributions in terms of the functional genes of the organisms. We have the environmental responses of these organisms with seasonal patterns and with our manipulations. So that gives us our functional genes and our environmental traits. And we would like to be able to relate those back to the decomposition. And we're looking at changes in plant polymer composition over this decomposition trajectory to relate those back together. We're also able to use this information to identify what traits are linked. So whether the possession of a specific enzyme is related to the possession of another enzyme. And that way, then, when you move to new environments, when you start to develop more information about how these traits are linked, when you move to other environments, you start to develop a consensus about what functions are typically co-located on genomes or co-housed within genomes. Then you can use that information without specific environmental information from a new environment to be able to parameterize or constrain these trait-based models. Okay. So to summarize, say we've developed this high-throughput functional metagenomics pipeline. And if you combine this with multiplex sequencing and sequence binning, you can then use this to constrain those enzyme trait distributions and the trait linkages necessary for modeling decomposition. And again, we're not relying just on annotation. We have biochemical confirmation of these, which is important. The next step then, though, is we have all of these genes. And one of the traits that we're still inferring and we're randomizing are the physiological, the kinetic properties of those enzymes. The kinetic properties and also their substrate ranges. And you can constrain those distributions based on literature values, but how well do those literature values represent the activities of these enzymes from this environment? And this is where really high throughput characterization of enzyme activity and substrate range, substrate activity is extremely important, and that's, I think, is the next step. But this needs to be higher throughput, so there's several technological hurdles to be overcome to get to that point. And then finally, one question that always kind of keeps us awake is really what fraction of the secretome, the enzymes that are secreted into the environment, are expressed in E. coli? How much of the picture are we missing? We know that we didn't see laccase, for example. We know that there are genes for laccase present. More than likely it's being expressed. Why is it not being expressed in E. coli? Can we get it expressed in another host? So we're working on that. But this really needs to be addressed on a much higher, at a much higher level. If we're trying to keep up with the growth of sequence information, our ability to biochemically confirm activities is way behind. So it needs some technological advances, and something potentially such as the microfluidic activity gel assays combined with proteomic sequencing, I think may be the way to do this. And if anybody here works in that area, I'd love to talk to you. Okay. So with that, acknowledgements. Most of this work was performed by a great postdoc in my lab, Mari Nisanen, with bioinformatics support from Ulas Karawas. We did our screening at the Joint Bioenergy Institute. We did some sequencing with Adam Arkin's group, and support from JGI for sequencing, and also Hudson Alpha for phosmid sequencing. The project PI is Steve Allison, and this is the rest of the team on the project, and funding from Genomic Science, DOE. Thank you.
Okay, I think we have time for one or two questions here, Matthias.
Very nice talk, Ian. I'm wondering if for your enzyme assays, if you actually lyse your cells, or if you lyse your cells, or if you don't.
Yeah, that's a good question. We use relatively long incubation periods, so we do get cell lysis, but we don't specifically lyse the cells. But we validated all these assays with pure proteins to detect to work on our sensitivity, and we grow these for 3 days, and after 3 days, you do get cell lysis and protein release. So it's not just about transport of the protein. Yeah.
Okay. Oh, Eddie has a question. I'll let him.
You know, the field of metagenomics began with functional phosmid screens, and it moved away from it because it was so low throughput. And so, did you ever think about transcriptomics or proteomics rather than screening for a very finite set of categories of functions? And
yeah, I mean, I think they're complementary rather than exclusive. You know, all we have here is a confirmation of functional potential. We don't know that these genes are being expressed in the environment. I think a perfect complement to this would be doing proteomics along the same time. Another advance would be doing CYP proteomics, where you have a stable isotope label substrate and you can trace that isotope into the proteins. That would provide another linkage in confirming the function of that protein in that activity. Another linkage. It's not proof. I think it's extremely important to also try and characterize these proteins. We really have no idea about the kinetic properties of these proteins or the substrate ranges. You know, we're screening on a relatively limited number of substrates just because of logistical constraints. But so, as I said, you know, we're doing this still kind of the old way, 96 well plates, we're pooling, we're trying to overcome some of those hurdles just by combining things and multiplex screening. But this really, to get to the next stage, this needs to be ramped up to, you know, using much lower numbers of reagents and much higher multiplex capabilities that microfluidics approaches can bring. Yeah.